Hello everyone. Welcome to our webinar titled Troubleshooting in Vitro Expansion of Leukemic Cells. My name is Sneha Balani and I'm a product manager at STEMCELL Technologies. I will be your moderator for this webinar. It is my pleasure to introduce you to our speaker. Dr. Leon Lin is a research and development scientist at STEMCELL Technologies in the Hematopoietic Research Group. He received his PhD, which focused on investigating novel therapeutic targets and microRNAs in chronic myeloid leukemia, from the University of British Columbia under the mentorship of Dr. Cheyenne Jiang at Terry Fox Laboratory, BC Cancer Agency. Since 2016, Leon has worked at STEMCELL, developing culture systems and assays tailored specifically for leukemic stem and progenitor cells. Let me now hand over to Leon to begin the presentation.
Oh, thank you, Sneha. And thank you for joining us for this live webinar today. So where I'm going to be troubleshooting in vitro expansion of leukemic cells. This is the brief overview of today's webinar. So firstly, I'll be giving some background on the myeloleukemia, and I'll be introducing some of the current common challenges facing the leukemia research today. And then I'll be moving on providing some of the solutions for culturing of leukemic stem and progenitor cells, as well as the development of our new product, the cell culture system. And then I'll be moving on to the next topic. I'll be talking about the application of our this cell culture system, the liquid culture-based drug screening assay. At the end of the webinar, I will be answering some of the questions that were submitted both prior and during the webinar. Okay, so let's now move on to the current common challenge faced in myeloid leukemia research today. For some background, so myeloid leukemia is a type of blood cancer. It can be further divided into a lymphoid lineage as well as a myeloid lineage. So each lineage can be further divided into a chronic phase or acute phase, depending on the percent of blood cells. Acute myeloid leukemia is the subtype that is the most lethal. It has the worst prognosis. CML without proper intervention can also progress to AML. Acute myeloid leukemia is believed to be a disease of aging, so the incidence rate is a lot higher in the developed countries. It is about 1 to 2 per 100,000 people in CML in North America, and it's about 4 to 5 people per 1,000 people in North America. So this translates quite well to the number of the publications. So number of the publications in AML is about 4 times higher than that of the CML. And in terms of patient survival, in CML, the patient survival has been improved quite drastically since the hydroxyurea and chemotherapy era, with the development of imatinib, which is able to target the BCR-ABL fusion gene. The overall survival has been improved to up to 85% in CML as compared to 30 years ago, where the patient survival is merely 11%. For AML patients, on the other hand, the patient survival has not improved too much since the 70s. You see this slight improvement of the patient survival is mostly due to the better patient care system. This is mainly because AML patients still mainly rely on chemotherapy today because the AML is highly heterogeneous. It's highly heterogeneous. It still remains a big challenge to develop a molecular therapy that is able to target AML. So even today, both CML and AML, the only curative therapy is still allogeneic stem cell transplantation. So myeloleukemia is a clonal stem cell disorder. CML will develop when the HSC, a hematopoietic stem cell, acquire a fusion gene BCR-ABL, and this will lead to the transformation of HSC into a leukemic stem cell, or LSC. The leukemic stem cell will then give rise to progeny, which will eventually replace the normal cells residing in the bone marrow. So clinically speaking, CML is a biphasic disease. It is defined by a chronic phase, or CML-CP, and blast phase, CML-BP. So chronic phase patient is characterized by overproliferation of the mature granulocytic cells, At this stage, tyrosine kinase inhibitors are quite effective in treatment of CML patients. The TKIs are quite effective in eliminating mature granulocytic cells. However, patients could acquire additional mutations beyond BCABL to progress to blast phase, which is characterized by overproliferation of the myeloid blast cells, and also in some patients, lymphoid blast cells. So at this stage, CML patients share many similar features as the AML patients, and the tyrosine kinase inhibitors are quite ineffective in eliminating these more primitive stem progenitor cells. So CML has been long served as a model disease because of its simple BCR-ABL fusion gene, its simple genomic abnormalities. AML, on the other hand, it's a lot more complicated. Different AML patients could harbor different genomic abnormalities, and even within the same patient, leukemic subclones carrying different genomic abnormalities could also exist. So for instance, in this patient, some of the subclones could respond to chemotherapy, while some of the subclones will not be able to respond to chemotherapy. And those leukemic clones are usually residing in the more primitive stem progenitor cells. That is why the current research areas in leukemia today heavily focus on identification of novel treatment as well as strategies that are able to target leukemia stem and progenitor cells. Because those are the populations that are responsible for drug resistance as well as disease relapse. Okay, now let's move on to some of the current challenges faced today in ex vivo culture of leukemia cells. It is actually quite difficult to culture those leukemia and stem progenitor cells or primary cells in general. For one thing, currently there's no standardized culture system for culturing those leukemia cells. So each research group has its own cytokine, in-house cytokine formulation, and cytokine formulation has different concentrations and combination of cytokines. Some research groups, they like to use interleukin-3 to promote leukemia cell proliferation, while some research groups, they use very little amount of IL-3 or no IL-3 to preserve leukemia stem and progenitor populations. Secondly, it is quite well known that leukemia stem and progenitor cells, they differentiate quite rarely in ex vivo culture. So in as little as 3 to 4 days, the major population of CD34 positive cells become negative. So this makes it actually extremely difficult for researchers to perform genetic manipulation for 3 to 4 days with lentiviral transduction, and it's very difficult for them to perform some of the downstream experiments, including drug screening as well as mouse transplantation models. Also, sample-to-sample variation is high. Some patient sample were able to grow in culture, while most of the patients actually do not survive quite well in ex vivo culture. And for the patient samples that actually do survive in culture, they don't tend to grow quite well after 7 days of culture. And the doubling time is extremely long. Patient sample is also quite limited. We all know that primary leukemia patient samples are able to recapitulate the complexity of the disease, but samples are quite precious and limited. They do not expand as well as the cell lines. They could not expand indefinitely. So oftentimes, researchers will make aliquot cryopreserved samples, but thawing those samples will also further reduce cell numbers. There has also been a large body of literature indicating the usefulness of preserving leukemic stem progenitor cells by culturing with mesenchymal stromal cells. However, the co-culture protocol itself could be quite laborious, and an additional isolation step is also required to purify leukemia stem progenitor cells prior to any downstream experiments. Okay, so we actually be able to provide some of the solutions to address those technical issues. Let's now move on to our next topic, culturing leukemia cells. So stem cell technologies have actually developed several product lines that are tailored for expanding and maintaining CD34 positive normal human stem and progenitor cells. So STEMCELL is a serum-free medium that is able to support the in vitro culture of hematopoietic cells. And STEMCELL CD34 expansion supplement is an optimized cytokine formulation that has been shown to be able to selectively expand and maintain CD34 positive cells obtained from cord blood and normal bone marrow samples. The pyramidal indel small molecules have been shown to be able to enhance HSC self-renewal. So U1729 was originally identified from a screen of compound capable of enhancing HSC self-renewal from the science paper published in 2014. UN729 later underwent a structure activity relationship optimization, which will lead to the development of a more potent compound UN171. So both of the small molecules are able to promote CD34 positive CD45RA minus population in vitro in a dose-dependent manner. And UN171 was also been shown to be able to promote the expansion of long-term HSC, as shown in this graph that the engraftment level of human engraftment level on NSG mice is further improved compared to the DMSR control and the fresh samples. And we STEMCELLs are the only licensed suppliers for providing UN729-1721. So we tested this system in our cord blood samples. Some of these are our in-house data showing that the cord blood cells that are able to expand quite well after 7 days of culture. So with the addition of UN171, we are able to further promote the expansion of CD34 positive cells, as well as maintaining better the CD34 percentage. So we later validate this system in AML and CML samples. In fact, all those components can now be sold together as a kit. We call it the STEMCELL leukemic cell culture kit. So before I move on to showing our leukemia data in culture, I would like to first address some of the common issues that are related to sample processing. This is a typical experimental setup to process fresh samples. So to isolate CD34 positive cells from fresh, either from bone marrow or peripheral blood samples, I would recommend our EasySep human cord blood CD34 positive selection kit too. So this kit includes a Rosetta CD34 pre-enrichment cocktail, which will facilitate the depletion of lineage positive cells, including T cell, B cell, granulocytes, platelets, and red blood cells in the density gradient median lymphoprep. The SEPMET also simplified the use of lymphoprep. Upon centrifugation, the mononuclear layers in the plasma fraction could be easily pulled off, while the red blood cells and the granulocytes also are retained well below the SEPMET barrier. The mononuclear cells can then be further CD34 selected using the CD34 positive selection cocktail. So oftentimes, the leukemia samples are cryopreserved. So to isolate CD34 positive cells from cryopreserved PBMC or BMNC samples, the same selection kit too can be used. I would like to point out that fresh samples should be only frozen down after CD34 positive selection, or at the very least, after red blood cell depletion or mononuclear cell isolation. So to isolate CD34 positive cell from cryopreserved sample, I would thaw the sample quickly in water bath and add DNase directly to the samples, incubate for 5 minutes at room temperature, and without doing any washing, the whole mixture can be proceeded to EasySep directly to maximize cell yield. So you could add the CD34 cocktail directly to the mixture. So if you want to further improve the cell yields, I recommend doing 3 rounds of cell separation in the magnet. So this will suffice to increase cell yield. And to have a better purity, 5 rounds of EasySep separation is recommended. Okay, following the afford protocol, the CD34 positive CML and AML cells can be purified. These are the typical examples of processing cryopreserved CML and AML sample. So often, the AML sample will have a higher CD34 positive starting percentage than CML. So again, this is mainly because the AML sample has a higher percentage of blood cells in their circulation. Regardless, both samples can be purified to yield at least 80% of CD34 positive cells. And now those CD34 positive cells can now be used for downstream experiments. So this is our experimental setup for cell culture. So again, we obtain a purified CD34 positive cell using EasySAP. Then we'll culture them in 1 to 10,000 cells per ml. And then we'll expand them for 7 to 14 days. And we'll perform readout at day 0, day 7, and day 14. So for the readout, we perform FACS analysis to measure the percentage of CD34 positive cells, as well as the total cell number of CD34 positive cells. We also perform CFU assay, which is able to assess whether those expanding cells will still have the colony forming potential. So these are the representative FACS plot of 1 CML sample. We're able to show that the STEMCELL leukemic cell culture kit is able to increase or better maintain CD34 percentage with the addition of UN-171 compared to the supplement without addition of UN-171. So to put all data together, those are the data from 6 different CML patient samples. So on average, after 7 days of culture, the frequency of CD34 positive CML cells could be maintained at 80%. And then after 14 days, the CD34 percentage can be maintained at least at 45%. In terms of total cell number yield, the CD34 positive CML cell number could be increased 65-fold after 7 days of culture. So in other words, if we were to see 1,000 cells at day 0, by the end of day 7, you will be able to obtain 65,000 CD34 positive CML cells. And those cell number can be further increased 10-fold more after 14 days. The system is highly robust. All the CML samples we have tested, 6 out of 6 of them were able to expand in this culture system. And these are the example of 1 AML sample. Very similar to CML, addition with UN-171 is able to better maintain CD34 percentage after 7 days of culture. And again, these are the results from 6 different AML samples. Again, we are able to better maintain CD34 percentage at about 80% after 7 days of culture, and at about 65% after 14 days of culture. And in terms of total cell number yield, the AML samples, they do not expand as well as CML samples, but the cell number could still be increased at least 30-fold on average after 7 days of culture. And very similarly, they can be further increased 10-fold after 14 days. And due to the heterogeneous nature of the AML samples, not all of the AML samples we have tested are able to expand, but the majority of them, 6 out of 10 AML samples were able to be expanded in this culture system. So now to assess whether those expanding cells are still progenitor cells and if they can still generate CFU-derived colonies, we perform CFU assay. So CFU assay is commonly used to detect multipotential lineage-restricted progenitors. That is for the erythroid granulocytes and macrophage. It is the benchmark of in vitro functional assay to study hematopoietic progenitor cells. So to test whether our expanded cells will also be able to form colonies, here we collect the cells that have been expanded for 7 days and the cells that have been expanded for 14 days, as well as the uncultured cells for comparison. So we plate those cells in our method called H4435 enriched medium, and we incubate for 14 days, and we then image and count the colonies with our STEM vision. So as expected, the uncultured cells, the cells that have not been expanded, were able to generate CFU-derived colonies. And the cells that have been expanded for 7 and 14 days, they both were able to generate CFU-derived colonies. So this is our CFU data collected from 6 different CML samples. It expresses total number of colonies per input CD34 positive cells in log scale. So for instance, if you were to seed 1,000 CML CD34 positive cells, you were able to generate about, on average, 100 colonies. And those are the theoretical total number of colonies from cells obtained after 7 days of expansion and after 14 days of expansion. So the cells cultured in SPAM2 plus experience supplements were able to generate those colony-forming progenitors. And with the addition of UN1CD1, so agree with our CD34 surface marking staining, addition of UN1CD1 is further able to promote the expansion of the colony-forming progenitor cell output by at least about 3-fold after 7 days of culture, or at least about 10-fold after 14 days of culture. And then now you might ask whether those colonies are derived from expansion cultures, if they will indeed arise from leukemic cells, or they could also arise from the normal cells residing in patient sample. So to address this, we actually perform genotyping by doing single colony qRT-PCR. So we plug single colonies, we track its RNA, then we perform qPCR using primers that are able to detect BCR-ABL fusion gene. And we also use our qPCR master mix to facilitate the qPCR process. Indeed, most colonies generated from expansion cultures are BCR-ABL positive. For instance, we plucked 99 single colonies from 6 different CML samples, and 92 of them indeed carry BCR-ABL fusion gene. Likewise, the colonies generated from expansion culture also yield very similar BCR-ABL frequency as the uncultured cells. Similarly, the AML cells that have been expanded for 7 and 14 days were also able to generate CFU colonies. And the addition of UN1CD1 was able to further promote the expansion of colony-forming progenitors compared to without UN171. I would like to point out that AML colony types are actually highly variable among patients. So unlike CML samples, where most of the patients will generate both BFUE derived as well as CFUGM derived colonies, the colony types vary somewhat in AML patient samples. So some samples are able to generate both type of colonies, whereas some of the patient samples are only able to generate CFUGM derived colonies. And the BFUE to CFUGM ratio is also quite highly variable among different AML patient samples. So unlike CML samples, where every CML patient will carry BCR-ABL fusion gene, so the leukemic colonies could be easily identified from CFU assays. The genetic background of those AML samples are actually not known. So to investigate whether those colonies are indeed derived from leukemic cells, I would recommend perform a single colony sequencing to identify those genomic abnormalities. Okay, so we will move on to the next topic. So this is an application of the cell culture system for leukemia stem progenitor cells. The assay will be the liquid culture-based drug screening assay. So this is a typical experimental setup for the liquid culture-based drug screening assay. So here again, we would purify CD34 positive cells from either fresh or frozen PBMCs or BMNCs. Then we would plate 1 to 2,000 cells in 200 microliter of leukemia cell culture medium, plus drugs, and we'll incubate them to drug treatment for 7 days. So at the end of day 7, we will analyze the drug response by looking at the image place plate reader or using a flow cytometer. So this is a typical picture taken from an image-based plate reader. The cells are highlighted in green or red, depending on their confluency. This confluency is proportional to cell numbers, so we can use this confluence percentage to graph this cell growth. Can also monitor the cell growth. And indeed, as you can see, the CML cells in our culture system are able to expand quite well after 7 days of culture. And indeed, those CML cells were able to respond to dasatinib in a time as well as a dose-dependent manner. the CML cells could also respond to imatinib in a dose-dependent manner. So this image is actually taken at 7 days after the drug treatment in triplicates, where we have cells control and cells treated with DMS or solvent and cells treated with imatinib from low to high concentration of imatinib. As you can see, the concentration, as the concentration of imatinib increase, the cell number will reduce. So technically, you could look at you could look at the drug response at different time points. Here we focus on 7 days after drug treatments. From here, we are able to generate a dose response curve based on a percentage of control, which is generated by the plate reader reading the cell confluence normalized to control against the log concentration of the drugs. So IC50 values can be determined from those response curves. So IC50 value is defined by the concentration of the drug that is able to inhibit 50% of the cell growth. So in this case, dasatinib IC50 value is estimated to be 9.0 nanomolar, and the IC50 value for imatinib is estimated to be 1.2 micromolar. So likewise, AML cells can also respond to both azacitidine and doxorubicin, or those two selected chemotherapy drugs, in a dose-dependent manner. So again, this picture is a representative image taken at 7 days after drug treatments, showing in duplicate. Also, those are the cells control, cells treated with DMS or solvent, and cells treated with from low dosage of doxorubicin to high dosage of doxorubicin, and from low dosage of azacitidine to high dosage of azacitidine. And those are the IC50 values that can be estimated from those dose response curves. So we actually have performed flow cytometry also to assess the IC50 values and to study their drug response. The results are very similar. Different readout methods can actually be used in this drug screening assay. So now I want to point out one of the current challenges, perhaps the biggest challenge in drug screening study today is cell numbers, the limited number of the cell. So one typical commercial cryopreserved vial will yield about 10 to 100,000 CD34 positive cells after CD34 selection. So to generate an dose response curve to estimate IC50 value, one will require at least 48,000 CD34 positive cells. So if we were to perform triplicate for 8 conditions, if you see 2,000 cells each, you will need at least 48,000 cells. So from a good sample, you might yield 100,000 cells. Only 1 to 2 drugs can be assessed with this cell number. And because our leukemia cell culture kit is a robust system, which is able to expand CML cells 65-fold and AML cells 30-fold after 7 days. So if you were to see even as little as 10,000 cells at day 0, the end of day 7, you will be able to generate more than 500,000 CD34 positive CML cells and close to 500,000 of CD34 positive AML cells. So this cell number will allow a more complete experiment setup to test multiple drugs. But however, we don't know that for sure if those expanded cells are also useful for drug screening purposes. So to test whether these expanding cells can also respond to drugs, if they could also further be expanded in the drug treatments, we perform very similar experiments as the non-expanding cells. So here, besides setting out experiments for the non-expanding or uncultured CD34 positive cells, we took a fraction of the cells, we cultured them for 7 days in the leukemia cell culture medium. We call this the 7-day expanding cells. And a fraction of the cells were also further expanded for another additional 7 days. We call it 14-day expanding cells. And all those cells were used in the liquid culture drug screening assay. So this is the dose response curves of the dasatinib and imatinib on the same CML patient sample I have shown earlier. So data shown here are the non-expanded cells. So this time we actually perform flow cytometry to measure the total number of 70 minus cells relative to control against the log scale of drug concentration. So compared to the non-expanded cells, both 7-day and 4-day expanding cells generate very similar dose response curves, which in turn generate very similar IC50 values for both imatinib and dasatinib. Likewise, in AML patient, both 7-day expanded as well as 14-day expanded cells were able to generate very similar dose response curves, which would yield very similar IC50 values. This liquid culture-based drug screening assay also allow assessment of drug specificity between normal and leukemic cells. So here we plot all the IC50 values together against the 4 drugs. And we are able to show that the leukemia cells are actually more responsive to drug treatments. So data shown here are the pool data of non-expanded and expanded cells from 4 different AML patients, 4 different CML patients, and from 4 healthy donors. So for example, CML sample which were labeled in blue show that upon exposure to imatinib, it has lower IC values than that of its normal counterparts. Likewise, treatment with dasatinib, we're able to show that the CML sample also have a lower IC50 value than that of the normal samples. So in general, all the leukemia cells are able to be more responsive to drugs compared to their normal counterparts. So this liquid culture-based drug screening assay seems to be working quite well. But how is it compared to the current gold standard hematotoxicity assay, the CFU assay? So to address this question, we also set up very similar experiments using the non-expanding cells, cells that have been expanding for 7 days, and then cells that have been expanding for 14 days. And we play all those cells in CFU assays, H435 medium, plus drugs. And we could pay them for 14 days and wait for colonies to be generated, and we would image and count the colonies. So this is a typical example of one CML patient, sample that is able to respond to both imatinib and dasatinib in a dose-dependent manner. So again, the dose response curve can be generated and the IC50 values can be estimated. And those are the respective colony pictures responding to dasatinib. Likewise, AML sample also respond to both doxorubicin and azacitidine in a dose-dependent manner in CFU assay. So to compare the 2 assays, the assay of liquid culture drug screening assay as well as the CFU assay, we plot the IC50 values obtained from CFU assay against the IC50 values obtained from the drug screening assay. Again, the data shown here are the pool data of both non-expanding and expanding cells obtained from 4 different AML samples, 4 different CML samples, and 4 healthy donors. The IC50 values obtained using each assay actually show the quite positive, strong correlation with a Pearson correlation coefficient of 0.82. Again, the leukemia cells show that they are more responsive to drugs as compared to their healthy counterparts in both CFU and liquid culture assay. So for instance, if you look at the AML sample which are labeled in triangle, their response to azacitidine is higher than that of the response to is higher than that of the normal counterparts response to azacitidine. So the value both vertically and horizontally, the IC50 values were lower. Okay, so to summarize, the STEMCELL leukemia cell culture kit is a highly robust system. All the CML samples we have tested were able to expand, and more than half of the AML samples we tested were also able to expand. This leukemia cell culture kit is able to maintain high percentage as well as high yield of CD34 positive cells after both 7-day and 40-day culture for both leukemic as well as normal cells. The system is also highly reproducible, as when 2 alloquads of the same sample were put in culture in different experiments. They will both generate very similar percentage of CD34 positive cells as well as total number of CD34 positive cells. The leukemia cells that have been expanded in using leukemic cell culture media are also quite useful for drug screening purposes. So the expanded cells, they respond to drugs in both liquid culture-based and CFU assays, and these two assays also correlate. It is able to generate IC50 values quite similar to that of the non-expanded cells. And because of these similar IC50 values, the expanded leukemia cells can actually use to compare with the normal cells in terms of drug response to better assess the drug specificity as well as toxicity. And if you were to look into more further information, please refer to our technical bulletin and our PIAs on STEMCELL website. Okay, with that, I would like to end it there. I would like to thank you for your attention.
Thank you very much, Leon. That concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the written questions that were submitted before and throughout the presentation. Let's start with our first question. This was a question submitted prior to the webinar. Is it necessary to select CD34 positive cells from patient bone marrow, or is it possible to take the entire patient bone marrow to culture?
Oh, that is actually a good question. So it really depends on the type of samples. In CML samples, most CML samples, their blast percentage is quite low. So their CD34% is usually pretty low. I will almost always perform a CD34 selection for a CML sample. AML samples, on the other hand, is actually quite variable. So some samples might have higher CD34%, while some might have lower. I will usually perform a CD34 selection if the AML sample were to have lower than 50% of CD34 positive cells. But usually from bone marrow samples, the percentage of CD34 positive cells are quite high. So I probably will not perform a CD34 selection on those samples. And also some of the AML samples, their CD34 minus population actually also have cancer initiating activity. So it might also be interesting to study those CD34 minus fraction in AML samples.
Okay, thank you, Leon. The next question is kind of related to this one. Somebody's asked us if STEMCELL Technologies sells equipment for purification of CD34 cells before kits for expansion. I think they're referring to the EasySep product line. So if you can just once again mention which specific kit you used, that would be great.
Oh, right. So we use the core blood human CD34 selection 2 kit. I think the catalog number is also mentioned in the technical bulletin and the PIS. You can refer to that further.
Yeah, that sounds good. The next question is about the STEMSpan leukemic cell culture kit. One of the viewers have asked if this kit can be used for the expansion of cell lines.
Oh, so we do not actually have the direct evidence showing whether the kit will also work on cell lines, but I would speculate so because our SFAM2 media does include serum substitutes. I would speculate that the kit should also work on the cell lines.
Okay. The next question was about the mechanism of UM171 in maintaining the CD34 cells. I think I can chime in here and ask the viewer to refer to a paper by Ferris et al. From Science in 2014. It's from Guy Savigeau's group. You can read that paper and you should be able to find the answer to your question. The next question is about drug responses. So a viewer has asked if you, during the drug response experiments, do you also culture them with the small molecule UM171?
Correct. UM171 is also added in the drug screening assays.
Mm-hmm. Thanks, Leon. The next one was a pre-submitted question where someone asked us, how likely is it that we'll get the same answer from a functional assay using cells if they've been refrozen and refrozen?
Oh, that's actually a good question. So yeah, so we occasionally would expand the cells and then we will actually freeze down before performing any experiments. And we found that those frozen expanding cells could also generate CFU-derived colonies, as well as they will have similar drug response compared to that of the expanded cells. But I wouldn't freeze down samples more than 2 times. I think one time it's okay. I will expand the cells and freeze down multiple aliquots. But if you're doing too many freeze and thawing, you will lose a lot more cells also.
Okay, thanks, Leon. The next two questions are pertaining to AML cells in particular. The first one is, why do you think the 4 AML samples did not expand, in your opinion?
So once you thaw this cryopreserved biosol, the viability varies somewhat among patient to patient. Some sample have very high viability and some might have lower. For those 4 samples that do not expand well in culture, perhaps their viability is lower, and also might be because maybe care wasn't taken when the vendors freeze down the cryopreserved samples, or could just be due to the heterogeneity of the AML itself, that some samples, because of their genetic background, some of the samples with certain genetic abnormal background simply could not expand in this culture system.
Okay, going off again on the AML cells, what is the maximum amount of time that you've been able to sustain your AML cells in active proliferation in our culture system?
Also, it also depends on the AML samples. But in the drug screening assay, where we have expand the cells for 14 days, and then we perform the drug treatments for additional 7 days. So that will be in total 21 days. And we found that the 4 of the AML samples we've selected are able to still expand quite well after 21 days of culture.
Thanks, Leon. There's also a question about CD34 positive, CD38 negative cells. So you've done most of your experiments with CD34 positive cells. Someone is asking, is it also useful for CD34 positive, CD8 negative cells?
So for the surface staining, we use CD34 positive, and we actually look at some of the primitive subset. So namely CD34 positive, CD45 RA minus, and CD30 90 positive cells. And we found that among those primitive subsets, using our culture system, we are also able to further improve the percentage as well as cell yield of those primitive population. As for the CD30 minus population, one could first sort with CD34 positive 38 minus population before they perform any cultural experiments. But CD38 marker might not be so useful to assess whether the cells are still primitive after culture. So most of the time, the CD38 population will become positive after culture. Might not be as useful as compared to other surface markers.
Okay, we'll end with one last question, which is a pretty broad question. How do you deal with contamination?
How to deal with contamination? I guess there are ways. Well, I guess I'm not sure if it's to prevent contamination or to deal with the contamination. Well, to prevent contamination, you would perform everything, have proper sterile techniques, do all the experiments in the BSC hood. I will autoclave all my tips. And then when I do mouse work back in grad schools, I don't tend to do mouse work and perform long-term culture on the same day. I found that sometimes mouse facility is not as clean as you have hoped. To deal contamination, so just thoroughly clean the incubators as well as the BSC hood with hydrogen peroxide and alcohol, and then throw away all the tubes that have been opened. Basically, a proper decontamination protocol should work.
Perfect. So the message is keep everything sterile. Well, okay. I think that's all the time that we have for today. I really want to thank everyone for attending, and I'd especially like to thank you, Leon, for your time and an excellent presentation.